My name's Nick Geiss. I'm the Chief Science Officer at a company called Curie Bio, based in Seattle, Washington, where it is not nearly this hot and humid, so it's been a bit of a week. We primarily focus on iPSC-derived models for drug discovery, and I'm going to talk about one of the things that we're working on currently, which is a contractile model of a skeletal and cardiac function. My first slide is kind of pointless to show because I think everyone has seen this probably about 10 times already, if not even today. But yeah, this is why we do what we do. And you know, again, we're just focused on making these human relevant models for drug discovery. As I mentioned before, we focus on muscle contractility. And muscle contractility is, affects everything. It's the reason why I'm able to stand up and my heart's beating right now. And there's a couple of different ways that we look at it. Number one is for safety. A lot of drugs fail in that big funnel that I just showed before because they screw up your heart. Drugs that have nothing, that were not designed to interact with the cardiac system at all will screw up cardiac contraction and cause problems. But also for discovery. And we can use iPSCs. I think there was a lady here who's asking a lot of questions about human iPSC models. We can use iPSCs to model diseases very easily, both in the cardiac and skeletal system. And contractility, to me, is really important to look at directly because contractility really results from a symphony of different biological subsystems working together to make this macroscale movement. You can have defects in the mitochondria that affect contractility. Defects in the cytoskeleton that can affect contractility, or ion channels, for example. A lot of different things need to work right to get contractility. So as we start to look for drugs that affect contractility, it's nice to have a system that encompasses all of this complexity in one go. And when it comes to gene therapies and skeletal models and things like that, the popular models are dogs and non-human primates, which not only are very expensive and cumbersome, but take a long time. And so that's why we're really focused on the iPSCs. So the platform we've developed is called MantaRay. And in this case, what we're doing is we're trying to make a turnkey system so that anybody can use these systems. I'm a cell biologist by training, and I know a lot of us kind of come from diverse backgrounds. There are a lot of bioengineers and folks who can do math. I can't do that. So when it comes to developing these types of systems, our benchmark is I need to be able to use it. And your average research associate at a drug company who's probably more from a biological background needs to be able to use these systems because one of the big challenges to doing these types of experiments is complexity. So this is the type of experiment we're trying to make more routine. Hopefully this pointer works. Basically, we make an organoid, which is a mishmash of cells and extracellular matrix on two posts. And you all have probably seen this. Casey's, of course, very famous system and paper that I've been talking about all week, so I'm excited to finally meet her. But also Thomas Eschenhagen did this in the early 2000s. And it's a really cool little system. This is a little it looks like a little noodle. It's about anywhere between 5 to 7 millimeters long, and it beats, it contracts. And you place this over two posts. One of these is deformable, and as this contracts, this post moves, and you can measure that delta. And from that delta, you can do a little bit of math and figure out how much force it's putting down. And you can do this over and over again as you add drugs or as you take away things or modify the genetics or even the cells used in here. You can see what happens. The problem with, as good as this system is and as useful it is to pharma folks, the challenge is this. This is a really good paper. I don't mean to knock anyone's paper. This is an awesome paper that used this type of model system to do some really cool biology. And this was the setup they were using. And it's really, really complicated. And trying to convince someone in pharma to say, hey, all you have to do is buy 20 different things and have a PhD level scientist spend a couple years hammering on this to get it to work, that's a tough sell. And unfortunately, I think what we've experienced is when we talk to the folks in pharma who try to do these experiments, they have a tolerance of about 6 months. And after 6 months, if they're not getting results, they move on. And typically, this is something it takes a couple of years to master. So we tried to distill this down into a much simpler system that's more turnkey and push button, if you will. Let me go back. Not only is this data collection very difficult, it's actually really hard to make these tissues for most people. The techniques aren't all that complicated. You can, just use simple pipettes and casting trenches and stuff, but we found that many people who have a couple years experience doing this will have a 50% or so success rate. And so what we did is we kind of saw these two challenges and said, okay, let's approach this. Number one, we started out as a cultureware company, so we know how to make highly reproducible cultureware vessels that'll assist people in making these types of things. And number two, we're also a hardware company, so we can actually build the kind of hardware to distill this down to a much more simple system. So starting out with building the organoid. So we set out to make a casting device to do this type of experiment. Has anyone ever made homemade popsicles in the summer? I do that with my kids all the time. Like, you get an ice cube tray, you fill it with fruit juice, you put a couple toothpicks in there, it freezes, and then you pull the toothpick out. It's the same thing. It's just much more expensive, unfortunately. So this is a cutaway of the casting tray that we make. Basically, you put your cells and your ECM into here, the fruit juice in here, and these two posts dip down into the fruit juice. It polymerizes, and you pull it up, and you get your EMTs. What we've done is we spent a lot of time optimizing the material selection and the geometry and dimensions here to make this fairly simple. We actually use a robot to do this in the lab. But we typically, will train a research associate who's fresh out of school with a biology degree, not much lab experience, to be able to get 95% success rate within a casting. So we can fabricate these things, and they're fairly reproducible. That's also one of the other challenges with making these types of organ-on-chip systems is reproducibility. If I get some data in January, I want to be able to compare it with something that I get next September using the same type of cells and the same type of techniques. So in these cases, we look at things like active twitch force, how much that little spaghetti noodle is contracting. And we also look at diameter, a geometrical thing. And we see, it takes a couple of days to actually stabilize these parameters, but once they do stabilize, we get a pretty good correspondence. These are different plates, so basically, this is a 24-weld plate, 24-weld plate, 24-weld plate, done over several weeks, and we can start to see after a couple of weeks we get good reproducibility, good stability when it comes to making these. So now we can start to do comparisons across time and across samples. When it comes to the contractility detection, the microscope technique is perfectly fine, but it's a bit cumbersome. You have to go to each tissue, take a movie. Sometimes you have to open up like a MATLAB or Python and do some custom programming to do the image tracking. We wanted to circumvent that, so we developed a magnetic sensor technology in concert with Nate Snydecki at the University of Washington, which is where we're based. And basically what we do is when you look at this thing, I think that's a movie. I don't know if that was playing while I was talking, but as this, John, I don't know if you can make that movie play again. As this soft post is moving, you see there's this black cube that's actually a very high-powered magnet, and that's kicking out a really big magnetic field. And this magnetic field can be detected several millimeters away. So underneath the plate, we put a magnetic sensor, and that magnetic sensor is outputting a voltage, and whenever it senses a change in the magnetic field, that voltage changes, and we can do some calibration in the factory so that every time we see that, we can actually very easily convert that into a distance and into a force using some very simple beam mechanics. I think you were showing some AFM talk or AFM images earlier, and much like AFM, we use an equation from the 17th century from Robert Hooke, so hardcore science, to actually get to a force of contraction. And the beauty of this is that we can do these calibrations and do these setups in our factory in Seattle and then have this thing just be ready to go. So all that you have to do is place the sample into the machine and it spits out contractility signals. And so we call this device the manta ray. Sorry, this is a shameless ad for my company. But it enables a very easy and rapid measurement of contractility. And because it's a non-destructive measurement, it's label-free, we can do longitudinal measurements quite easily. This is a cardiac safety example. This is a BMS 986094. So it's a drug you've never heard of because it never made it out of the clinic. It's a hepatitis C drug. So hepatitis C is the liver. and it was designed to help patients with hepatitis C, and it looked really good, and they put it into clinical trials, and it killed people. It actually caused heart failure in several of the patients that died. And we could have caught that. So this was in 2012 when this happened, so predates the iPSC technology that we rely on now. But we can see a dose-response-like fashion over the course of a couple of weeks. And so, in addition to acute drug data that we've been talking about quite a lot over beers this week, we're starting to look at chronic effects too. And you can see that as you escalate the dose, you start to see the dip in contractility, or the twitch frequency in this case, much earlier in a dose-response-like fashion. So it enables those types of experiments. From these, I think I stole this from Casey's paper, so I'll buy you a beer later for it. From these data, you can actually get a wealth of information. People like to look at things like frequency, rate of contraction, rate of relaxation. There's a whole laundry list, and this isn't even comprehensive. And the reason I put this up is that because as we start to look at disease models and different defective modalities in these tissues, it turns out not all of these are created equal. In some cases, the disease will only show up in frequency, or in rate of relaxation, or maximal twitch amplitude, for example. So we like to measure all of these things, chart all of these things out so we can actually find the hallmarks of disease. And I'll show an example of that in just a moment. So we're starting to focus not only on cardiac systems, but on skeletal muscle systems, specifically for discovery applications. In this case, we typically have been working a lot on iPSCs, but in this example, we're looking at primary skeletal myoblasts. In this case, this is an EMT that we've already done the experiment on, and of course, it's over. So then we can cryosection it and do the stains, and we can start to look at what proteins are there. So, for example, this is a skeletal muscle where we can look at dystrophin. Sorry, that's not projecting too well. And myosin, just to see that we have a nice healthy core inside the middle of the tissue. There's no necrosis that we can detect. But also we can look at specific expression of specific proteins. So for example, we had a customer who was very, very interested in GLUT4 because they wanted a diabetic model of skeletal muscle function. We're very easily able to probe those kinds of things and answer those kinds of questions in these EMTs with follow-on assays. The data kind of look like this. When we get the recording from the magnetic device, it plots it in terms of force versus time. We get nice twitches. So in this case, this is a stimulated primary myoblast EMT, where we're pacing this thing at 1 hertz. You can put electrodes into the machine and push a button, and you get this thing to actually twitch. But even more importantly, we're able to actually replicate tetanus and force frequency, which are hallmarks of maturity in skeletal muscle systems. If you've ever kind of woken up in the middle of the night with like a muscle cramp, which I did the other day, that's your nerve firing really fast. So fast that your muscle can't relax and you get into this tetanic response. And being able to do that is actually a good thing. It's a marker of muscle maturity. And we can start to replicate that here. And also, there's a force frequency effect too. So the faster you start pacing, the higher that force gets. And we do see that this is something I ripped from a paper, a very cool paper, where they measured this in humans. You can see this in a mature human muscle. We get the same kind of response in our EMT systems. Oops, sorry, that's a duplicate slide. So why are we doing this? Well, we want to see drug responses. So you can do more acute drug responses, as I mentioned before. Example with BDM, it's an ATPase inhibitor. It basically deactivates contraction. And when you put it onto these EMTs, you can see very clearly there's an IC50 of 3.2 millimolar. We see very clear response of reduction of contractility. Similarly, we can have activators. So this is a troponin activator, and we could do things like washing out. So in this case, we're able to give a bolus of the drug to the tissue and then wash it out and see fairly good recovery of the tissue after. So we can enable both acute and chronic experiments, as well as multiplex this with other assays to kind of find your favorite molecule of interest. In addition, we've been really focusing on disease modeling. So a lot of you may have heard of muscular dystrophy, and that's, dystrophin gets mutated and your muscles waste away. It's a really devastating disease. Typically, 30 years ago, you wouldn't have gotten out of your teenage years, or maybe you'd make it to 20. before you die. Now, with the standard of care, they're actually giving these patients ventilators, and they have portable ventilators that they can put in their wheelchair to keep them breathing. So when the diaphragm gives out, they can still live. However, we've been finding that a lot of these patients are now dying from cardiac dysfunction. So there's a cardiac phenotype that only shows up later in life, and a lot of the treatments don't affect that. So what we can do is, using iPSCs, we can start to model this on a chip. In this case, we have a wild type, they're both green buttons, it's bad design. There's a wild type iPSC line that we've got from a healthy patient, and then we knocked out the DMD, so we basically did a CRISPR engineered DMD version of this patient, and we can start to see the dystrophin disappears once you start to do that. And when we make cardiac EMTs, we can look at a couple things. Number one, amplitude very clearly is different, so it's a much weaker tissue that you end up making with this genetic defect. But also there's an arrhythmia. Normally these things pace by themselves very nicely, and you'll get a nice, fairly rhythmic beat. However, in the disease state, you get an arrhythmia, and that's typically what kills these kids. They'll go to bed one night, they have an arrhythmia in the middle of the night, and they don't wake up. So we can model this in the cardiac system, but the beauty of this too is that we can also model this in the skeletal muscle system from the same line. So in this case, these are, again, iPSC lines, and we can, and in this case, we're making skeletal muscle, and we see similar deficiencies when it comes to things like force. I mentioned that laundry list of metrics that we can measure that is still from Casey's paper, and you can see that the disease state shows up in most of them, but in some disease cases, many of these will be equal, and you have to kind of find the correct hallmark. But we can extract all of these kinds of data, relaxation velocity, contraction velocity, tetanus, things like that, from these types of contractility data. We can also measure fatigue. One thing DMD patients really suffer from is that their muscles fatigue very quickly. And as we stimulate these cells, we see that the strength wanes over time. In a healthy person that happens, but in a DMD patient it's much more severe. And this is just another example of that. Sorry, I thought I deleted this slide. And finally, now that we have, as Paul was saying, we have the assay windows that we can look at. Now that we have that, we can use this as a test for different therapies. So one of our big future directions right now is using this as a platform to screen AAV therapies, so gene therapies. And in that case, you're hitting the patient with an AAV that contains a transgene, that's the fixed dystrophin or the fixed gene of interest. And can you actually model this on a chip? Because right now your choices are mouse, monkey, or human. And some of these diseases you can't even model in a mouse, and a non-human primate study can cost upwards of $5 million and several years of work. So as we start to build these organ-on-chip scalable technologies, we can start to do these experiments. This is just an AAV with a reporter, a cherry reporter, showing that we can actually transfect these cells and show that the reporter gene is there and the construct is alive, and we can start looking at contractile efficiency and contractile hallmarks once we treat these cells. And then, I don't know how much time I have, but this should be a movie that plays. We can start to, as I said, multiplex other assays. We can start to do calcium. That's a movie that should play. Yeah, there we go. So, we can start to do excitation, contraction, coupling, and other things like that. We just released a new system. Again, another shameless advert, because my marketing guy's here and he'll yell at me if I don't do this. We've developed a new optical mapping system in collaboration with Todd Herron at the University of Michigan, where we can start to simply stick in a plate and start to do voltage mapping and calcium mapping. And the next generation of this is to apply those to these EMTs, so we could do these multiplexed label-free experiments. And so with that, I think, I was supposed to have a conclusion slide, but I guess I cut it. My time's up, but basically, at Curie, we're really interested in taking these very sophisticated 3D bioengineering experiments and making them easy and facile, so that folks in the pharma community can actually use them, and use these as models of not only toxicity, but of efficacy with disease modeling. So that's my talk. Thank you.